Hello friends, welcome to today's lecture. Please, if you haven't subscribed to this YouTube channel, do well to click on the subscribe icon. So for today's lecture, we'll be talking about the metabolism of low density lipoprotein. So in our previous class, we talked about the metabolism of very low density lipoprotein, where we discussed that the very low density lipoprotein particle functions mainly in the transportation of triglycerides to extrahepatic tissues or peripheral tissues such as the adipose tissue, skeletal muscle, and cardiac muscle. We also stated that when this very low density lipoprotein particle gets to the capillary wall of these various peripheral tissues, an enzyme called lipoprotein lipase is usually located, and this enzyme hydrolyzes a triglyceride contained in the very low density lipoprotein, and that will result in the formation of intermediate density lipoprotein. Now remember, very low density lipoprotein contains mainly triglyceride, but it also contains small amount of cholesterol esters, proteins, and phospholipids. So once this triglyceride has been hydrolyzed, it will result in the formation of intermediate density lipoprotein, which contains a smaller amount of triglycerides and then larger amount of cholesterol. But then this LDL particle contains ApoE and ApoB 100. So we stated in the previous class that this ApoE can be recognized by its specific receptor on the surface of the liver cells. And so if there is a recognition of the ApoE by its specific receptor, the LDL particle will be taken up by the liver cells. But if such recognition is not there or it does not occur, then ApoE will be given back to the HDL particle, which will result in the formation of LDL particle. What it means is the LDL particle contains only ApoB 100 and then mainly cholesterol. Also, on the surface of the liver cells, there are specific receptors for ApoB 100. So once this receptor recognizes ApoB 100, ApoB 100 can bind to this receptor, and that can cause the LDL molecule to go into the liver cells through a process of receptor-mediated endocytosis. In normal conditions, about 70% of the LDL particle is taken up by the liver cells, and then inside the liver cells, they are metabolized and then they are excreted through the bile duct into the small intestine. And then only about 30% of these LDL particles are taken up by extrahepatic tissues. But for the purpose of this lecture, we'll focus on how the hepatic cells can take up these LDL particles. This is a diagram which represents a hepatocyte or the liver cell. Now, this area represents the extracellular space, which is interaction with the blood circulation, while this represents the intracellular space. So this is the inner portion of the liver cell, while this is the outer portion of the liver cell. Now, on the surface of the hepatocyte, there are these specific receptors for LDL particles, which are negatively charged glycoproteins. So they are widely dispersed on the surface of these hepatocytes. But then, before LDL particle can be taken into the liver cell, two major proteins will swing into action. The first is the adaptor proteins. So what the adaptor proteins do is, the adaptor proteins are present on the surface of the liver cell. And what they do is, they stimulate these receptors to accumulate at a localized region. So instead of dispersing, instead of being spread around every part of the liver cell, these adaptor proteins ensures that these receptors move to a particular location or a particular locality of the liver cell, as well represented there. They accumulate at this point. Now, the second one is the clathrin protein. The clathrin proteins. And this clathrin protein, unlike the adaptor proteins, which are located on the outer portion of the cell membrane, these clathrin proteins are present on the inner portion, okay, the inner portion of the hepatocytes, somewhere here on the hepatocyte. And what these clathrin proteins do is it causes an invagination of the cell membrane where these receptors have been accumulated. So the first is the accumulation of these specific receptors at a particular locality or particular location of the liver cell by adaptor proteins. And then the second one is clathrin proteins, which causes the invagination, okay, which causes an invagination of the plasma membrane, that part of the plasma membrane or that locality of the plasma membrane where the receptors have been accumulated, as shown here. Now, this is the area where the receptors have been accumulated, and then clathrin proteins will cause an invagination of this area. So this represents the LDL particle bound to their specific receptors. Now, this can also be called clathrin-coated pit because this coating or this invagination was induced by clathrin proteins. Now, this invagination will further lead to the formation of vesicle. Okay, so in the vesicle, we can see these receptors also bound to the LDL particles. And then we have the clathrin proteins. These are the clathrin proteins. So these clathrin proteins are also present here. Now, after the formation of the vesicle, the clathrin proteins will be removed from this vesicle and then it will be recycled. That is, it will go back into the liver cell to be reused for further synthesis. So the removal of the clathrin proteins will result in the formation of endosome. Now, in the endosome, we have the receptors, and then we have the LDL particle that are separated, that have been separated from the receptors. And how does this happen? The endosome has an ATP pump, okay? So it hydrolyzes ATP and then it moves hydrogen ion into this part of the endosome. It moves hydrogen ion into this part of the endosome. And once an hydrogen ion goes in, it will reduce the pH of the medium, of course, which will create an acidic pH. Now, this acidic pH will then result in the dissociation of the LDL particle from their specific receptors. Here you can see they are bound to the receptors, but here, because of the presence of this hydrogen ion as a result of this endosome, these LDL particles will be separated from their specific receptors. Now, before I proceed, there is an enzyme called PCSK9. PCSK9 means proprotein convertase subtisylene hexene type 9. What this protein does is, or what this enzyme does is, it binds to this complex. Usually, it binds to this complex, and then it prevents the dissociation of LDL particle from the receptor. Also, it leads to the hydrolysis of this receptor. It means it breaks down these receptors such that the receptors cannot be reused. So this is just an introduction of PCSK9, but then I'm going to come back to it. Now, after the dissociation of this LDL particle from their specific receptors, it will result in the formation of this. So now we can see a portion that contains only the LDL particle, and then another portion that contains their specific receptors. Now, this portion containing only the receptors is called the call. What is call? Call means compartment for uncoupling. Compartment for uncoupling of receptor and ligand. So this call will further dissociate to form a vesicle on its own, and these receptors will be recycled. It means they will move back into the plasma membrane, okay, or to a cell membrane, and then be reused or be recycled. Also, this portion containing the LDL particles will fuse with lysosomes, and because lysosomes have the hydrolyzing ability or hydrolyzing potential, they will break down these LDL particles to form amino acids, fatty acids, phospholipids, and cholesterol. Now, this process takes place over and over and over again. And then when it gets to a point where the amount of cholesterol inside the liver cell is enough or is high, you can see this sign shows that an increased concentration of cholesterol. Once there is an increased concentration of cholesterol in the liver cells, there must be a feedback inhibition to say that we do not want more cholesterol to come into the cells. What we have is enough. And so we do not want more cholesterol to come into the cell. And also, we should also remember that cholesterol is also synthesized by the liver cells through a process of what? De novo synthesis or endogenous synthesis. So in the liver cells, cholesterol is synthesized, and then cholesterol also come into the liver cells from the circulation. So there are three basic regulatory mechanisms that will occur once there is an increased concentration of cholesterol in the liver cells. First is that excess of this cholesterol will be converted to cholesterol esters, and that will be catalyzed by an enzyme called ACAT. And what is ACAT? ACAT is acyl-CoA cholesterol acyl transferase. So this enzyme will convert the excess cholesterol to form cholesterol esters, which will be stored in the liver. So the cholesterol is stored in the liver in form of cholesterol esters. The second inhibitory mechanism or regulatory mechanism is that this increased cholesterol will stimulate a decreased production of HMG-CoA reductase. Now, this enzyme functions or stimulate the endogenous synthesis of cholesterol. That is the synthesis of cholesterol by the liver cells. So once there is a decreased concentration of this enzyme, it means there won't be sufficient synthesis of cholesterol by the liver cells. And then the third regulatory mechanism is that increased concentration of cholesterol will stimulate the decreased production, okay, and recycling of LDL receptors. And this actually makes sense because if there is sufficient amount of cholesterol in the liver cells, it means the liver cells do not have to synthesize more cholesterol because what it has is enough. And so it will inhibit this enzyme which functions in synthesis of cholesterol. So once this enzyme is inhibited, it means its liver cells cannot produce cholesterol. And because the amount of LDL particles that come into the liver cells is dependent on the number of its specific receptors on the cell membrane. That is to say, if there are more receptors on the surface of the liver cells, more LDL particle will go in. But if there is little amount of receptors on the liver cells, it means only small amount of LDL particle can go into the circulation. And so because the liver cells do not need more cholesterol, it will also inhibit or cause a decrease in the expression of the LDL receptors. Now, I would like to come back to the PCSK9. Now, certain disease conditions can actually stimulate an increased concentration of PCSK9. I stated earlier that PCSK9 functions in breaking down these LDL receptors because usually, immediately they are being used there, they are recycled. It means they go back to the cell membrane or to the surface of the cells. But when the level of PCSK9 in the liver cells is high, okay, when the level of PCSK9 in the liver cell is high, it means more of these receptors will be broken down. And so only few amount of receptors will be present on the surface of the liver cell. And when few amount of receptors are present on liver cells, it means few amount of LDL particle can go into the liver cells, and then the bulk of the LDL particle will remain in the circulation, which will pose a risk for atherosclerosis. And one of such conditions is hypothyroidism. So hypothyroidism is a condition where the thyroid gland cannot produce sufficient amount of hormones. So hypothyroidism stimulates an increased production of PCSK9, and an increased production of PCSK9 will result in an increased hydrolysis or breakdown of LDL receptors, which will further result or lead to a decreased expression of the LDL receptors, which will in turn lead to a larger amount of LDL particles remaining in the circulation and taken up by extrahepatic tissues, which also can lead to atherosclerosis. Now, another thing I'd like to talk about are scavenger receptors. Now, scavenger receptors are receptors present on the surface of our macrophages, and these macrophages flow freely in our circulation. So, LDL particles, because they are referred to as the bad cholesterol, LDL particles, while flowing, most times get oxidized, especially in capillaries. Let's assume this is a capillary. This is a capillary wall. This is a capillary wall. And then there is an LDL particle here. Okay? And then let's assume this LDL particle becomes oxidized. And then in the circulation, there are macrophages, okay, that flow freely. Now, immediately this becomes oxidized, the macrophages from the circulation will flow into the capillary and engulf it. Okay, it will engulf it, which is actually a good thing. But if this process occurs repeatedly over and over again, it will cause an inflammation of the wall of the capillaries, okay, which over time will result in the deposition of atherosclerotic plaques on the walls of the capillaries. That particular place that inflammation occurs, there will be deposition of atherosclerotic plaques, which in turn will lead to atherosclerosis.